Okay, welcome again to the new era of Biogeography Symposium. Thank you for being with us again. So our next speaker is Dr. Fabricio Villalobos from Evolutionary Macroecology Laboratory and Evolutionary Network from Ecology Institute from Mexico and Laboratorio de Ecologia Teórica e Síntese Departamento de Ecologia Universidade Federal de Goiás do Brasil. He works in macroecological theory and methods, integrating macroecology with phylogenetic approach to understand geography patterns of biodiversity. His talk is entitled Species, Features, and Evolutionary Macroecology Approach to Study Biodiversity Patterns. The talk will be in English, but he will receive questions in English, Spanish, and Portuguese. Thank you, Dr. Fabricio, for your talk at our symposium. Welcome. You can continue now. Thank you very much.
Thank you, Oscar, and thanks to all the organizers for the invitation. Let me share my screen. There, can you see the?
Yes, we can.
No? Okay, excellent. So thanks again for the invitation and the opportunity to tell you a little bit about my research, especially about this idea that I have been working on with some colleagues for the last 10 years now, actually, about the species fields, a concept and a conceptual tool that we have been developing to study biodiversity patterns. And I want to show a couple of examples of how to apply this particular concept to study geographic biodiversity patterns. So we all know, and most of us know, that life on Earth is varied, you know, biodiversity, and we have a lot of species, a lot of life forms, but also that biodiversity is not homogeneously distributed on Earth. And we can see this classic pattern, which is the latitudinal diversity gradient, where we have more species in the tropical regions compared to less number of species in the temperate regions. And this pattern has been tried to explain in different times with different hypotheses. But we know that on the back of this pattern, what really causes is the distribution of the species. And not all the species are distributed everywhere, and not all of the sites have the same number of species. So we have this heterogeneous distribution of life on Earth that actually depends on the distribution of the particular species. So especially in biogeography and in macroecology, we want to know what causes that distribution of species and the co-occurrence of those species to form this particular type of geographic biodiversity patterns. So we need to focus on the particular unit of a study, which is the distribution of the species, right? So we would like to study the distribution of the species and use it as a study unit to link interspecific patterns or interspecific patterns or whole assemblages patterns, considering particular biotas or particular regions or the whole planet, right? And we can study the area of distribution by looking at the form or the size of the distribution. For example, sorry, let me put the laser pointer here. There we go. We can study the form and size of the distribution, if it's circular, if it's irregular, or something. We can use actually fractal geometry. We can study the structure of the distribution of the species in terms of morphology or genetics. For example, we can measure the genetic diversity of a particular species across the distribution, across its distribution, or the size of the body, body length or body size. Or we can look at the location within a particular geographical gradient or environmental gradient, or the concurrence of those species. And we're going to be talking particularly about the structure of geographical ranges. And usually that structure is studied in terms of intrinsic traits of species, like body size or its genetic diversity or abundance, for example, how many individuals you have in different parts of the range of a particular species. But here I'm going to argue that we can study the structure by looking at other species, by looking at one particular species and study the structure of that species, but by looking at the distribution of other species. So let me explain that particular idea, which we term the diversity field. So we have here a geographically biased and taxonomically biased example. Within Mexico, we have a distribution of a particular bat species, here an endemic bat species of Mexico. And that species co-occurs with other species of the same genus or related genus in the family Phylostomidae, which are distributed across Mexico and South America. But we actually want to know what's happening with that first species. So we can look at that species, the first one, the Glossophaga morenomi species, and actually describe a richness gradient, for example, based on the co-occurrence of other species within the distribution of my focal species, right? And I can do that for all of the species, of course, but I can do that one by one. And from that, I can derive different metrics. For example, just a simple frequency distribution of how many sites have different numbers of species. And in the case of these species, for example, just as an example, we have most of the sites. Here's the sites on the y-axis, and here are the number of species on the x-axis. Most of the sites have low number of species, and less number of sites have higher number of species. And we can use that to characterize each species. We can think about what we call the diversity field, right? The co-occurrence of other species within the range of a particular species. And here I'm just putting 5 particular examples that go from species that co-occur with a low number of species. They don't like to co-occur with other species, in this case, particularly bats, and with this particular bat, which is the tequila bat. But we have other species that co-occur, for example, this one down here, that co-occurs with a lot of other species. So we have like a distributional footprint of that particular species, a tendency of the species to occur in a species rich or a species poor regions, or in particular center of origin, or cradles or museums, and we're going to discuss that later. Okay, so we can use this concept of the diversity field as an species property and think about the tendency of species to occur in particular regions, and we can use it as a footprint, as a particular signature of the processes behind that co-occurrence. And we can think about how does it look, what are the causes of that. Or we can even go further and think about the geographic template as John Thompson's mosaic of coevolution theory. We can think about potential interactions, morphology, community assembly, and particularly phylogenetic relationships, which is very common nowadays, that we have large numbers of phylogenies available at the species level, etc. Right? So, including that phylogenetic information, there's a field, a subfield of ecology, which is called community phylogenetics approach, which uses phylogenetic information to understand the assembly of communities. And we have 2 main alternatives of patterns. One is the negative relationship between phylogenetic distance and co-occurrence, which says that species closely related in the phylogeny, they inherit traits and they can be in the same communities, and that's a phylogenetic cluster pattern. And the alternative is a positive relationship of co-occurrence with phylogenetic distance, and that's the phylogenetic over dispersed pattern, that you have species from different lineages. And usually that type of approach is based on sites, on local sites, and you have a community. And our argument here is that we can use that to study whole ranges, right? So if we export that idea to the distribution of the species, to the whole distribution of the species, we can think about the structure, the phylogenetic structure of a species within a particular focal range, right? So for example, we have this species here in yellow, and we have these other species that occur with it across the distribution. And those other species, they can be distantly related, right? Over dispersed pattern, or they can be closely related, or they can be randomly related, right? So we can use that to study the co-occurrence of species and associate that with life history traits, and think about the try to infer some particular processes. And that's what we have been doing, for example, when we try to couple phylogenetic information and morphological information that can tell us something about ecology and evolutionary processes, as people have done before, use that the degree of species co-occurrence with the degree of similarity, morphological similarity or phylogenetic similarity to study geographic diversification. We can use those two and put it in the context of the focal species approach, or a species field approach, which I'm calling it here. And that's what we did a few years ago with my friends and colleagues. We looked at the global patterns of mammalian co-occurrence, considering phylogenetic structure and morphological structure. And what we found was that something that we already knew, but not at the level of species. We have two main histories, two main stories, evolutionary stories in the assembly of mammalian communities across the world. We have old lineages in the old world and newer lineages in the new world, and we see a clearly longitudinal pattern of new world, old world phylogenetic co-occurrence. And when we look at body size, the traits of the species, that the pattern is completely different, right? So we have species that co-occur with similarly sized species, with species of similar body sizes over here in the north, in the temperate regions, and in the tropical regions, we have species that co-occur with other species that are more different in terms of body size, right? And what we can extract from that, and we use some new models, and I invite you to read the paper if you're interested in, but we consider some biogeographical pools, the history of the biogeographical scenario, and we show that most species are random in terms of the phylogenetic fields, but that actually tells us something about the different evolutionary histories between the old world and the new world. And also, we looked at significantly clustered phylogenetic fields and over dispersed phylogenetic fields, looking at phylogeny, but also looking at body size, and those species were present in different regions. The clustered ones and the over dispersed ones were present at different regions, which suggests that the effect of evolutionary processes are different in those particular regions. And we can pinpoint some regions that are more interesting to study, right? So, and another important thing was that the phylogenetic structure was similar, was congruent with the results from the trait field, from the body size field that we were looking at. So both things were telling us the same in terms of body size or phylogeny, which is not very surprising because we know that body size has a strong phylogenetic signal. But that also tells us that probably ecological processes such as competition or character displacement are not that important at the large scale. And just for another example, how we can use this idea of the species fields or the phylogenetic fields to study some other aspects of biological patterns. For example, in terms of biological invasions and something known as Darwin's conundrum, which is the idea that Darwin had that exotic species, to invade a particular native community, they have to be similar to those natives, right? So that's the pre-adaptation hypothesis, that you have some characters or some traits that allow you to be introduced in a particular native community. But then he found out that actually the species that invade other communities, the exotic species that invade other communities, they're actually from different lineages. So that became known as the naturalization hypothesis, which makes both make different predictions, right? So that has been termed Darwin's conundrum. And this idea of the structure of communities considering exotics and native species has been studied at the level of sites also, looking at particular communities, looking at particular regions. And our argument here is that we can use the species field approach to study that. And we have two main properties. We have the invasiveness of the species, the tendency of the species to be more, to be able to invade a particular region. And we also have the invasibility of sites, the tendency of sites to accept exotic species. And with my good friends from the University of Ryukyus, my Japanese friends, we published this paper a couple of years ago using the phylogenetic field approach to reconcile the Darwin's conundrum, naturalization and pre-adaptation. And we came up with this theoretical scenario that we have four extreme scenarios. We have one scenario where phylogenetically similar exotics colonize phylogenetically similar natives, a cluster recipient, right? Or we can have phylogenetically similar exotic colonizing an over-dispersed community, native community, or we can have a phylogenetically distinct exotic colonizing a cluster recipient, right, of the closely related natives, or we can have both a phylogenetically distinct exotic and a phylogenetically distinct community, the recipient community. And what we found out was that both properties, invasiveness, the species invasiveness, the capacity of the species to invade, and the invasibility, the capacity of sites to accept new species, are positively related. And we have different patterns. We have or 3 or 4 scenarios over here, but the two most common scenarios were the extremes, the first and the fourth scenario. We found out that the exotics are related with natives. They colonize assemblages of natives that are themselves related across the recipient. And we also found that exotics that are distant to the natives colonize assemblages of natives that are themselves also distantly related. So we can, so we found out that pre-adaptation and naturalization are both possible, and they were identified by applying the species-based approach, the phylogenetic field or the species field more generally to these particular questions. Of course, we can add more information, and that's what my good friend Jesus Pinto Ledesma did a year ago when we published this paper in Ecological Monographs, testing the Darwin's conundrum, but we use taxonomic, phylogenetic, and functional dimensions of vascular plants, looking at the co-occurrence of exotic species with natives, considering their phylogenetic structure and their trait structure, considering specific leaf area and other traits of plants. And what he found out, I'm not going to show all the details, but what he found out was that the non-natives or exotic species tend to occur with closely related natives, mainly across the whole time. The functional structure doesn't change, but the phylogenetic structure changes. He did that in a classic experiment of Cedar Creek in Minnesota, which have data for 40 years and a fire gradient. And but the idea here was to use the focal species approach and include also functional traits. And he found out that those things are telling us different things. The phylogenetic field and the functional field, they tell us different things because the functional field doesn't change, but the phylogenetic field does change. So that's an idea that we're trying to put out there and to study different patterns using this species field approach. And stay tuned, because soon, hopefully, my student Alejandro Sanchez Barradas, which is doing his PhD with me at the lab, he's studying the cross-trophic field, trying to integrate trophic interactions, mainly predator interactions, in this idea of the focal species approach. And with that, I just want to say that we can use the species fields as a tool for macroecology and biogeography, as a species-oriented perspective to study biogeographical patterns, as a tendency of species to occur in particular regions. We can include biogeographical processes, evolutionary, ecological ones. We can think about the invasion dynamics, or nowadays we can think about interactions at the broader spatial scale. And with that, I want to thank the organizers and lots of friends and colleagues and students, and of course funding agencies, and you guys for inviting me, and also for listening to me. Thanks.
Thanks, Dr. Fabricio, for your talk. It was very interesting and amazing. Now we have time for some questions from the audience, okay?
Sure.
There are 2 questions in Spanish. So, the first, in Spanish. Entonces, considera que la accesibilidad geográfica de las especies, M del diagrama BAM de modelado de nicho, podría definirse a través de inferencias respecto a las relaciones bióticas de las especies B en el diagrama?
Es una pregunta interesante porque junta dos líneas de investigación. Yo no me dedico tanto al modelado de los nichos, pero soy colaborador con especialistas de esa área, muy grandes amigos, y hemos discutido mucho esta cuestión de la accesibilidad, ¿no? ¿Cómo defines? Que no es una cosa fácil de hacer. ¿Cómo defines esa accesibilidad? Y la idea que nos preguntan aquí es que si podemos usar eso para definirla y usar las interacciones, las inferencias bióticas, ¿no? La concurrencia con otras especies para definir esa accesibilidad. Yo creo que sí se pueden usar esas inferencias, pensar en dónde están presentes las especies con las que nuestra especie focal concurre, y entonces pensar en esas posibilidades bióticas de que la especie esté presente. Esa es una posibilidad. No la hemos explorado, pero es una buena idea adicionar esa información de inferencia biótica para llegar al área de accesibilidad. Excelente idea.
Vale, ok, gracias. La siguiente pregunta en español es, ¿encontraron relación entre tamaño corporal y algún factor extrínseco, por ejemplo, latitud o temperatura?
Encontramos relación entre tamaño corporal y un factor extrínseco. Con tamaño corporal sí encontramos una relación latitudinal más tradicional. en donde las especies, mamíferos en este caso, de las especies de mamíferos, y eso se cumplía para diferentes órdenes de mamíferos, en la región tropical coexisten con especies más diferentes en tamaño. Hay una mayor diversidad en tamaño y las especies coocurren con conjuntos de especies que son más diferentes en tamaño, ¿no? Entonces se están repartiendo el espacio ecológico con más especies. Ya las especies que están en regiones templadas lo hacen con especies más parecidas en tamaño. En ese sentido, pues podemos pensar en los mamíferos grandes, ¿no? La mayoría son grandes en región templada, entonces todos coexisten con organismos más grandes. No hemos explorado las cuestiones ambientales, no sabemos si eso va a estar relacionado con temperatura. Podríamos pensar que sí, porque hay una asociación con latitud, pero sabemos que esto es más de una dimensión, entonces no lo hemos explorado en ese sentido. Gracias.
Okay, well, the next question is in English. So
Okay.
the first question is, does species fields only limit to mobile species, or it can be used for studying the flora composition?
Excellent question. Thanks. Of course, yeah, it can be used to study flora composition. Actually, the idea of Darwin's solving or trying to solve Darwin's conundrum came from using plants as a study system. In the paper that I showed for my Japanese friends, they have an excellent database of the Japanese flora, and that's where we tested the idea, using Japanese flora, considering natives and non-natives. And my friend Jesus Pinto Ledesma used the Cedar Creek experiment with plants to study that idea of reconciling Darwin's conundrum using this focal species approach. So, of course, yeah, what we need is the occurrence of a species, the geographical ranges or the sites where species occur, and from then we can apply this idea, yeah.
Okay, thank you. The next question is, do you think it's possible to infer the chains of invasion based on the phylogenetic structure of a community?
I do, yes. I think that we can use that. Actually, that's an excellent idea, because you can try to infer if a particular community has more invasibility, has a tendency to accept new species as exotics, if you know where they come from in terms of lineages, right? If they are closely related, if they are distantly related, or if they don't care about the relationships, you can use that idea, and based on our results, for example, for the Japanese archipelago or for the Cedar Creek experiment, we can not safely, but we can go further and say, okay, so if particular communities have a cluster composition, for example, they are all closely related, the exotic that may invade that community has to be closely related. So we can say that, for example. And if there's a community that is composed by distantly related lineages, the exotic that is going to invade that community, hypothetically, has to be distantly related also. That's what those two extreme scenarios that we found. So yes, it is possible to infer some chances, the possibility of invasion based on those phylogenetic or functional structure, looking at particular species. So the idea here is to go out of the site or the assemblage approach and start looking at the species approach, looking at the distribution of the species based on other species co-occurrence. That's the whole idea. Yeah.
Okay, there isn't more questions.
Okay.
These topics are so interesting. I think maybe we need to do other talks in another symposium, maybe, I don't know. Okay. Okay, again, thanks, Dr. Fabricio, for your talk and your support in the symposium and until a new opportunity. Thank you very much. And now we invite you to connect to the next talk. Remember, to be interesting. Okay, thank you, Doctor. Thank you very much. I said, remember, who be interested in be part of the staff from the next symposium in 2022, you can write to the general email from SBE. Okay, no, don't forget. Thank you very much. Bye-bye, everyone.